Museum. Allison has a long affiliation with MVC. She was one of our star undergraduates here. She graduated in 2007, and while an undergraduate, she worked on the Grinnell Resurvey project. She did her honors thesis looking at how bird communities have changed over time on the campus here. And then after graduating, she was hired by Craig to coordinate our centennial celebration in 2008. Allison, after that, went on to get a master's with Kevin Burns at San Diego State, and then from there went to Harvard, where she's getting her PhD with Scott Edwards. And working with Kevin and Scott make her Joseph Grinnell's academic great-great-granddaughter, because both Kevin and Scott got their degrees from Ned Johnson. And anyway, Allison is here actually preparing specimens for her research. She wanted some house finches, and she volunteered to come out and actually prep them. So I talked her into giving a MVC lunch on what she's been up to, and she's going to talk about, I guess, tanagers and house finches. So it's great to have you back, Allison.
All right, well, thank you for that introduction. It's really great to be back. You know, it's kind of like coming home. It's been quite a long time since I've been here, but it's been really great to see so many friendly faces. So I just had, last time I was up here giving this talk, I was talking about birds of the Berkeley campus, and I've come a long way since then, since thinking about kind of how communities have changed on a very small scale. I've kind of become interested in, what actually drives this huge diversity of life that we see. And so how, in an evolutionary context, have all these different animals and plants and other creatures come into being. And so one can address these questions from a few different scales. For example, one can look at it on a very grand scale, looking at whole big groups of organisms. One also can address this on a smaller scale, looking within one species and how populations might differ both phenotypically and genetically over their range. Or even on a very short time scale, in even 30 years, and similar to what we've seen here with the Grinnell Resurvey project. So for my research, I'm going to start on a very grand scale, and I'm going to start with looking at the evolution of sexual dimorphism. And so sexual dimorphism, which is when a male is going to have some sort of ornaments. For example, they can have a very bright plumage or bright ornament that's going to be a product of females choosing more brightly ornamented males, or these ornaments can be due to male-male competition. And in the past, the evolution of these characters have been thought to have been caused by changes in male characteristics. So what do I mean in that? For example, you might have a species that starts off with two drably colored males and females, but over time, selection for a brighter male might cause bright male coloration. So that is going to be what I'm calling male-driven changes in sexual dichromatism. And dichromatism, that's just changes or differences in coloration rather than changes in overall morphology. However, one can end up with that same phenotype by a completely different mechanism, and that's if you start off with two brightly colored sexes, a bright male and a bright female, you might have actually natural selection on the female to become more drab that can lead to that same phenotype. And so there have been a number of studies that support both of these avenues for the evolution of sexual dichromatism in birds. And so for my own study, I wanted to look at this in tanagers. Now, tanagers are an excellent group to study this. About half of them we think of as being monochromatic, which means that males and females have the same plumage pattern. About half of them are dichromatic, and you have everything ranging from very bright males and females all the way to very drab males and females that have the same plumage, to where you might have a bright male and a drab female, or have fairly drab males and females but that have different plumage. And so the amount of variation on this family, which has about 370 species of birds, is an excellent opportunity to use phylogenetic comparative methods to understand how sexual dichromatism evolves. Now, I'm not the first person to think of this, so my master's advisor, Kevin Burns, in his PhD, when he was actually here at the MVC, addressed this in a genus level. So what he did was he categorized different genera of birds as being bright males, bright females, bright males, drab, bright males, drab females, etc. and found that actually in tanagers it seems to be more driven by the evolution of female plumage than the evolution of male plumage patterns. But there are a few different reasons that the study needs to be redone. There's a lot of variation in genera of tanagers, and as you'll see in a minute, they're actually not very monophyletic, so the named genus of a tanager is not a very good way of categorizing how their plumage is. And also, this is using the human visual perspective, and as we know, birds don't see the ways that humans do. And so, a little bit more about tanagers. They comprise, like I said, 371 species and about 10% of passerines or songbirds. They have a huge amount of diversity, both in plumage coloration, but also in morphology and song, and they live all throughout Central and South America. So the first thing that you need to do when you want to conduct a comparative study like this is to make a phylogeny of your animals. And so I kind of jumped into this when it was in the middle of when most of the data had been collected but still needed to be analyzed. And so what we ended up having was sampling 355 of the 371 species of tanagers, with 4 cardinal species as an outgroup. The cardinals are the sister group to tanagers. 6 genes, 2 mitochondrial, 4 nuclear, and using a Raxmel and Beast approach, we constructed a phylogeny of these guys. And what we found was that there are 15 subfamilies of tanagers that have radiated essentially at the same time at the base of the tanager tree. So the base of the tree is a big polytomy where these different groups have come from. And they range greatly in the size of species, anywhere from about 100 species in the Thrauponae subfamily that we designated, which is a lot of the really beautiful tanagers that one thinks of, like the Tangara species or the mountain tanagers, all the way down to single monotypic subfamilies. Now, thinking about diversification, one thing we wanted to know was, is this an example of an adaptive radiation? And so we made a lineage through time plot where you essentially count how lineages are accumulating through time. And so the dotted line here represents what you would expect if lineages were just being accumulated in a clock-like pattern. And we found that we have a pretty large signal of adaptive radiation where you get a lot of lineages accumulating earlier and then slowing down through time. Now, we also used the BAM method developed by Dan Roboski to see if there are any shifts in diversification rate over the group. And what we found were there were 2 major signals of diversification shifts, 2 big increases in diversification. One was in Darwin's finches, and so that was really nice to see since a lot of people considered Darwin finches to be an adaptive radiation. But as far as we know, no one's actually shown that in the context of their evolutionary history, but also in the subfamily Sporophyllinae, which are sporophyllous seed eaters. So back to tanager dichromatism. As I said, birds have a different visual system than humans, so let me just, for those of you that don't remember off the top of your head what that is, let me go into a little bit more detail. So as humans, we have 3 cone types, and we designate 400 nanometers wavelengths of light to 700 as being the visible wavelengths of light, and that's because that's essentially what we can see with our 3 cone types. Now, birds actually have a 4th cone that is activated by 300 to 400 nanometer wavelengths of light, and so that's essentially in the UV part of the spectrum. So birds can see UV colors that we can't see. They're also, so because they're tetrachromatic, they're seeing colors in essentially a 3-dimensional context rather than a 2-dimensional, which is what we see. A little bit more on that in a second. And so you might wonder, how important is this UV coloration in tanagers? But it actually is very important. So this is an example of one species, the glistening green tanager. And they have these, kind of grayish patches on their wing coverts and on their ear patch. And if you actually spec them, you can see this is a reflectance spectrum. So how much light is being reflected across the wavelengths of light. Most of the reflectance in these patches is in the UV spectrum. So this is essentially a UV colored patch. So if you could actually see UV colors, this would be very bright and not kind of the just dull grayish patch that it looks like to us. And so if we think about, how can we quantify these colors? Okay, so you have a reflectance spectrum, but let's think about, how can we quantify them with the Avian visual system? Like I said, so as humans, we've got these three cone types, short, medium, and long wavelengths. And you can take a color and plot it somewhere in the middle based on how stimulated each of these are. And as I said before, that's kind of, you could think of it as being in a two-dimensional space. But because birds have this fourth UV cone type, not only is it in the two-dimensional space, but there's an additional dimension to color that we simply can't see. So just, a lot of people tell me, okay, so can you make something that, how do birds see this? How can you demonstrate to me what that would be like? And of course, I can't. There's no like bird goggles that you can put on. It's been suggested to me that I create some. But so for example, if we take this painted bunting and we take out all the red, so if we had no red cone, this is what we would see. So you can see there's quite a bit of variation that you miss from not having that cone type. Now in addition, they also have these oil droplets that are on top of each cone cell. And so light has to go through these oil droplets before the cone cell is actually activated. So this is a picture of a bird retina, and the cone cells, the oil droplets are colored with carotenoids that are specific to the type of cone cell. And what that means is that they're actually absorbing the excess light that is not necessary for that particular cone cell. So it's going to be activated a lot more specifically. And so what that means is that birds are not only going to be able to see this extra color, but they're going to be able to differentiate between very subtle colors a lot better than we can. And so as I said before, we think of birds as being about half dichromatic, half monochromatic. And so one of the first things that I did for my thesis was to quantify sexual dichromatism from the avian visual system. So I took measurements from all of the different patches on males and females of the tanagers and ran them through the avian visual model and got a value which is what the unit is called JNDs, or just noticeable differences. And a value of 0 to 1 means there's no difference, but anything above 1 means that there's actually a distinguishable difference between the two colors. And so when we think about it, if we actually plot, okay, so what's the level of dichromatism in each species, where actually 97% of them fall above this cutoff line of 1. And it's not just that the monochromatic species, while they are more highly clustered towards the lower levels of dichromatism, there are some that are actually of pretty high values. So there really are actual large differences that we simply aren't capturing. And so going forward, I wanted to study how is sexual dichromatism evolving, and so I couldn't just look at these specimens and classify them as colorful dichromatic, colorful monochromatic, as Kevin had done many years ago. And so I used an Ocean Optics 2000 spectrophotometer and pulsing on light source, which is kind of your standard reflectance spectrometer for measuring plumage coloration. I measured up to 5 females and 5 males per species that were collected from the same locality and time period, and measured 9 standard patch regions plus any additional color patches that I saw. And so that actually was up to 16 color patches in some of the really colorful Tangara species. Each measurement was repeated 3 times, and in order to do this, I got to do a grand tour of many of the museums around the country, including the MVC. So I was here to measure some birds, that was probably 5 years ago or something like that. In total, I collected 90,207 reflectance spectra over about 30,000 plumage patches from just under 3,000 individuals from 361 of the 371 species. So it was a pretty complete data set. And so in order to, I used the tetracolor space model in order to in order to actually analyze these reflectance spectra. And so just to walk you through how this works, so this is Chlorocrescent intedissima, which is the 7-colored tanager. So for example, if I was going to measure the back, the breast, and the belly, the reflectance spectrum might look something like this. And basically what you do is you overlay how stimulated each of the different cone types are by the reflectance spectrum. With this, we can convert it into 3D coordinates that go into the avian tetrahedral color space. And so in order to actually measure sexual dichromatism, what I did was I did the same thing for males and females, and then quantified dichromatism as the difference, or the distance between these points in the 3D color space. And I took the dichromatism measurement as the largest measurement between males and females, because birds see each other on a patch-to-patch basis, not necessarily as an average of all of their patches. So, historically people have thought of color in terms of hue, chroma, and brilliance, and so you actually can get measures that are related to these things. So for hue, we actually have two measures, theta and phi, which are the angles. So if there was the achromatic origin, which is at the center of the tetrahedral color space, you think as latitude, or the angle out to the vector, that's where so if my patch is right here, there's a vector from the achromatic origin to the patch itself. And so theta is going to be the latitude to that vector, and phi is going to be the longitude. One thing to note is that theta is mostly made up by the short, medium, and long wavelength color cones, whereas phi has a very big ultraviolet component to it. R is going to be what we're going to be calling chroma, and so that's the distance from the achromatic origin to this patch, or the length of that vector. And then brilliance is actually calculated independent of the tetrahedral color space, but it's just average reflectance over the course of the whole spectrum. And so I did this for all of the color patches for males and females that I had taken, and actually have a package that if you can find on my website to do this, called R-tetracolor space. It's an R package. And so from this you can take a number of different measurements that can actually represent aspects of the entire plumage. So color span is going to be the average distance between all of these different patch measurements, and so that's going to be representative of overall plumage contrast. Hue disparity is going to be the average and distance between the hues of these things. Color volume, this is, I think, kind of one of my favorite measurements, but that's the volume of the minimum convex polygon fit to those points, so you can think of that as how colorful the bird is. Average chroma, and then also average brilliance. And so, okay, now here's the question. So I've got this data set, now what can we do with it? Again, my first question was, is the evolution of sexual dichromatism in tanagers driven by changes in female plumage coloration, as Kevin found many years ago? Second, I'm going to ask, have females and males evolved under different selective pressures? And finally, I'm going to pull in some information from one of my lab mates on tanager song and ask, is there a correlation between song and male plumage complexity? So for our first question. So to do this, I used a phylogenetic generalized least squares approach. I did all this in R. And so basically what I did here was, so I took all of the standard patches that I had measured across all of my species, so that's 9 different patches, and for each of those I calculated theta, phi, R, and brilliance, and I used stepwise ASC to fit the best model to look for a correlation between sexual dichromatism and then these patch measurements for both males and females, so a separate model for both. Oh, sorry, I actually had 12 plumage reasons I forgot. because I'd been able to extrapolate it out to a few regions that I'd measured across most of my individuals. I had 336 species with genetic data, male measurements and female measurements. And I used Pagel's lambda for my phylogenetic correlation matrix. And so what this graph is showing is the difference in AIC score between these models for the males and the females. So blue bars, so this is basically the difference between how well these models fit, and blue bars are going to be models that fit better for males, and red bars are going to be models that fit better for females. Gray bars are going to be models that had a difference less than 4, which is not, you can't really tell the difference between their models, so you could say they both fit about the same. And so one thing to note here is that actually most of these regions had a better fit to the male models, so correlation between sexual chromatism and male plumage measurements, which is different from what Kevin had found those many years ago. Now there were a few that fit better with female, but it was interesting to note that those are plumage regions that I wouldn't necessarily associate with being particularly important in signaling, like the dorsal tail and the tips of the wings, and they might actually be under more natural selection. The exception to this is the wing coverts, so I was kind of surprised to see that the female model fit better for that, so that's like these brightly colored epaulets that a lot of species have, but those could be important for female signaling as well. And so this is what I found across all tanagers. And so I was like, okay, great, that's a nice story, right? But what happens if we look at some of these subfamilies we found? So we see this pattern across all tanagers. Does that hold up when we break it down into some of the major subfamilies? And the answer is no. Interestingly, for most of these subfamilies, actually there are a lot more female correlations than male correlations. And so what does that mean? It means that overall there's some signal that you can only detect when you're looking across all tanagers, but perhaps these different subfamilies have different things going on with their life histories. And so generalizations that you can make across, you know, a big family of tanagers actually might not necessarily be appropriate if you're only looking at a very small subsample of that. And so I decided to look at this in a different way as well. So I took I took these overall whole plumage values that I had calculated before and ran them through a phylogenetic PCA, so I could get a measure of plumage coloration that kind of incorporated all of these different measures. And they all loaded pretty heavily onto PC1 except for brilliance. And so I decided to use that PC1 as kind of plumage coloration. A high value towards 1 is going to be something that has very kind of what we would think of as colorful plumage. There's a lot of different color patches that are all quite contrasting with each other. And the value towards the more negative the value, the more drab that they are. And so again, I looked for a correlation between this PC1 and dichromatism for males and females, and found that there was only a correlation with males. And so that does support kind of what I found on a patch-by-patch basis. I don't have the slides in here for all of my different subfamilies, but again, that was a bit of a mixed bag. Some had a correlation with females and some had a correlation with males. So not as clean-cut of a story as we might like, but you know, that's the way biology kind of is sometimes. And so my next question, how have females and males evolved under different selective pressures? And so to do this, I used some different models. So this is across all tanagers. What I did is I took my several measures, average color span, color volume, and hue disparity, and chroma and brilliance. And I asked, do they fit better a model of Brownian motion, which is just random evolution across the tree, a model where there's different selective optima based on if individuals live in enclosed or an open habitat, and the most complex model, which was habitat plus foraging stratum, so that's where the closed habitat was divided up by canopy and understory, or just is there a single optimal phenotype, plumage phenotype for these things. Now why did I choose habitat as something to look at? Well, you might imagine that depending on what kind of habitat you lived in, there might be, for example, there might be different types of coloration that might make you more cryptic or more conspicuous. And so, for example, in an understory, if you're a dark color bird, you're going to be quite cryptic, but if you're in the canopy, if you're a dark bird, you're going to stand out more, and if you're a light bird, you actually might be more cryptic. So you might actually have different selective pressures operating in these different habitats. And so what did I find? I found that for both males and females across all of the measures, the most complex model that included habitat and foraging stratum was the most favored. And so that suggests that habitat is actually quite important in driving the evolution of these different plumage characteristics. And so again, that was across all tanagers. And so what happens when we look at some of the different subspecies? So for all tanagers, again, habitat and strata. For Puspizinae, which was another separate study that I published, I found that for, sorry, this is males. Males had habitat as the best fit. Females had habitat and Brownian motion kind of as equally competing models as the best fit. But when you look at the rest of the subfamilies, again, we get all of these different models that fit best depending on where they are. So again, I think this group is so diverse that there's not really one single model that you can apply to the whole thing. And so my final question for this is there a correlation between song and male plumage complexity? So I kind of joined forces with my lab mate Nick Mason, who's looking at the evolution of song in this group. And so you might wonder, why was I looking for a correlation between song and plumage complexity? There's actually a rich history of study of this question. And even in Darwin's time, he had allusions to transference of them, in that, you know, you don't usually see bright colors and bright songs in the same species. And so the idea is, if it's very expensive to have bright plumage coloration, or very complex songs, you might see one or the other, but not both at the same time. Now, previous studies have demonstrated a mixture of results. Some have found this, and some have not. And so for Nick's song dataset, he had collected just under 3,000 songs from 321 species. From these, he quantified 20 frequency and temporal characteristics of the song using the program Raven. And again, we wanted to get kind of a measure of plumage complexities. We used phylogenetic principal component analysis and used PC1, where the more negative the PC scores had a larger bandwidth, but that means that they kind of sang at overall different frequencies. They had more frequent dramatic frequency shifts, and that's something that we kind of consider to be a complex song character. They had more notes, greater note count, more different kinds of notes, and a longer song. So overall, the more negative the PC score you're going to have, the more complex your song is going to be. Now, I already told you about the same thing that we did for plumage coloration, but again, we used PC1, and so in this case, the more positive you were, the kind of more colorful you were. And so just to give you an idea of what a complex song sounds like, so you see there's a lot of different shifts, a lot of different notes that we listen to. And then, a second here. Now here's a simple song. So it's very short, there's just a few notes, they're not as different of shifts. So things that fell out as having very complex plumage are some of these species here. Things that fell out as having very simple plumage are some of these species here. So our PC scores kind of, you know, even though we can't see as birds do, they fit very well into what you would think of as having very complex plumage. This is just showing that again. Same thing in the tetrahedral color space. Something that has very complex plumage has a very big color volume, a very large range of points, whereas simple plumage, they're all kind of clustered in one area. So again, we used phylogenetic, generalized least squares. We had a data set of 303 species that had all of the different data types. We looked for 9 different song plumage correlations, and so we did these different PC scores and then a couple specific plumage and song scores that had been used in previous studies. We compared AICs that had a base model that included subfamily as an interaction character, habitat type, the presence of carotenoid coloration, and the presence of structural coloration. So we were trying to take into account all of these variables that might actually be correlated with this sexual dichromatism, with song and plumage complexity. And again, we used phylogenetic correlation matrix scaled by Pegg's lambda. And so what we found was that habitat type, carotenoid coloration, structural coloration did not do well compared to either just a simple model or a model that included subfamily, and that of these, none of them were significantly correlated. So we found no correlation between complex plumage and complex songs. What you see here with these were the correlations that actually the best model fit with different subfamilies, and so each of these lines is the correlation for that subfamily. And you can see there's no single trend. Some families go one way, some families go another way. But again, these correlations generally, they were not significant either. So in conclusion to the tanager part of the talk, as a whole, the evolution of sexual dichromatism in tanagers is driven by the evolution of male plumage coloration, although this kind of breaks apart when you look at it subclade. There's changes in both male and female plumage coloration. Light environment is important in shaping plumage evolution for both males and females. And in tanagers, song and plumage elaboration are largely uncorrelated. Okay, so now, I'd looked at these questions, looked at evolution across a very broad scale, across a large family of birds. Now, what would happen though if we looked at, one specific species of bird? And so, for my dissertation, that's what I decided to do. I went away from the tanagers and decided to look at the house finch, which is not a tanager, but it's a finch, which is somewhat closely related. But house finches are very important because they are very interesting for several demographic reasons, as well as for studies of disease ecology, sexual selection, and they're becoming a model of organism for many different fields. And so just a little bit about the house finch. So males have this kind of bright red or yellow or orange plumage. Females are kind of just brown and streaky. And they were made pretty famous because they're one of the first times that Jeff Hill in 1991 showed that sexual selection and this condition-dependent plumage trait are actually correlated. So males that are yellow have the lowest female preference, whereas the redder you are, the more females will prefer you. And that's actually related to carotenoid coloration, which has a number of interacting terms, including based on diet. So you have to actually eat things that can give you this bright red coloration. Now, some of the basic house finch demographic history. So they're native to the western US and Mexico. There's a number of different subspecies, which are each of these little polygons in different kind of overall species types. But for our purposes, I'm going to focus on the frontalis subspecies. So that's the one that we generally find in the US. And so, what makes these things interesting is that in 1870, or at least sometime before 1870, they were introduced to Hawaii and now are very common there. Actually, I had to point out that it was Joseph Grinnell who first wrote about this back in 1911 when he had gone to Hawaii and had found house finches common on all 4 Hawaiian islands. So, have to go back to my kind of Grinnellian roots in all avenues of research. But also in 1940, they were introduced to the East Coast, to the New York area. And this happened because they've been popular as pet birds. They have really nice colors, they have nice songs. But in 1940 or so, the International Migratory Bird Treaty Act began to be enforced and it became illegal to have them as pets. So what happens when they're illegal to have as pets? People release them into the wild. So by 1960, they had a bit of a foothold on the eastern US. There's a paper written in 1953 that thought that there might have been a bottleneck in as few as 20 individuals in this area. 1970, we see expansion and kind of progressing to where now they're virtually everywhere on the East Coast, and there's also some kind of interesting admixture of the two different populations. And so we have this bottleneck followed by a very big range expansion in the East, which is interesting from a kind of looking at how this type of demographic event can shape genetic variation in a species. So what makes them even more interesting is that in 1994, this Mycoplasma gallisepticum bacteria, which is associated with poultry and actually a big problem in the poultry industry, jumped to house finches in the Washington, D.C. area. Within about 2 to 3 years, it had spread through the entire eastern population and killed off about 60% of the individuals. So it had a very large effect on the eastern house finch population. They still have not recovered from this disease. They're still about 60% of what they were in the early 90s. In about 2002 or so, the disease jumped to the western population, where it's been much slower to spread and less prevalent, but is actually gaining in virulence. But there are still a few areas that it hasn't spread to yet, and we have no idea what's happening in Mexico with it. And so some previous folks in my lab, I'm not the first one to study this system, have done some interesting experimental infections where they took individuals from the Tempe, Arizona population, which has been historically unexposed, it's never seen mycoplasma before, and individuals from Alabama where they had been exposed to the disease for about 10 years, and experimentally infected them and found that the Alabama population had a lower mg load compared to the Arizona population. So they seem to have developed some sort of resistance to the mycoplasma. They also looked at gene expression across a number of genes and found that there were quite a few genes that were upregulated in the Alabama population but downregulated in the Arizona population. One thing that the MG is thought to do is actually suppress a host's immune system. So both of these things together suggest that there may have been some evolution of resistance in the house finch due to this strong selective event. There have also been subsequent experimental infections using these same populations, which measured some inflammatory signaling molecules and found that individuals in Arizona seemed to be more tolerant than individuals in Alabama were more tolerant than individuals in Arizona, and which supports again the evolution of not only resistance but tolerance to this disease. And so for my PhD, I want to study the genetic basis of this resistance and tolerance as well as the demographic founder event. So to do this, I'm going to be sampling. I'm going to conduct several different comparisons. So the western population versus the eastern population. I'm going to look at how I have samples that were collected before the epizootic event, so I can look at just how this demographic event, bottleneck plus expansion, affected the genetic diversity of this eastern population. And then by sampling through time both of these populations as well, the same populations, I can look at how then subsequently their genes have changed due to the mycoplasma. So to start with, I'm going to focus on the demographic event. And so I'm actually not the first person to study this either. Some people in Scott's lab and also folks at Cornell have looked at this. And so there have been some support using AFLP data, RFLP and Alzheimer's, that there was no change in effective population size in the introduced Hawaii and eastern populations compared to the native western population. However, Dana Hawley at Cornell used microsats and ND2 and found actually that there was a reduced effective population size, about 30% of what they previously were in the eastern population. And so that begs the question of, what is actually the answer? One difference is that in these studies, she used individuals collected after the epizootic event, whereas the previous study had used individuals before the epizootic event. And so was that decrease in genetic diversity a consequence of the epizootic event or a consequence of the founder event? And so I decided I wanted to explore that question. And so to do that, I didn't sample them because they're collected in 1990 before the epizootic, and I was not a biologist at that point in time. But across the range, our collaborator, Alan Baker, had decided in 1990 to do a phylogeographic study of the house finch. And he did this in 1990 right before the mycoplasma, which is very convenient for me. And so each of these points represents a frontalis population that I had samples from. I sequenced 3 to 6 individuals per population. I also had 4 individuals from this Grisconii subspecies as an outgroup, and several purple and Cassin's finches, which are the sister species. And so what I used here was double digest RADseq, which I know people in this museum have been using and have heard of. I used EcoR1 and SPHI enzymes, and so I sequenced about 15,000 loci per individual. I used 150 base pair paired and HiSeq 2500 sequencing. At the time, we didn't have a house finch genome, so I used both bowtie2 to map to the zebra finch genome, which is about 7% divergent, and also tried a de novo assembly. I called variants using the STAX pipeline, and then ran a number of different PubGen analyses, as you'll see. And so, just to remind you guys about double digest versus single digest RADseq. So in single digest RADseq, you have an enzyme that cuts the genome, and then you sequence from both directions of that cut site. And with that, you can actually build kind of longer fragments, and you have reads that will be spanning both sides of the cut site. With double digest RADseq, you're cutting the genome with 2 different restriction enzymes, and you're going to be sequencing from both ends of that fragment. Because you're going to have reads that stack up because they're always anchored by a restriction site on either side. Again, I said I use STAX for de novo assembly. Now, one thing about STAX with this double digest RADseq data is that it can't really handle the paired end data as a single fragment, because the stacks have to line up with each other exactly. And so right now your options, if you have double digest data, paired end double digest data, is to run each end separately or to concatenate them into one big file and totally disregard which end of the fragment it came from. And this is perhaps problematic because if you have SNPs that are from the same end of your 300 base pair fragment, they might be in linkage to equilibrium, and so they might bias your results. So you've got things coming from both ends. So I actually wrote a number of different scripts that will actually repair your loci after running them through STAX. If any of you are interested in doing that, feel free to talk to me at some point. But basically, you build stacks just as normal, and then after the fact, you can not only see if sequences that were sequenced on the same fragment always align, so you always get, okay, stacks from this end of the fragment always align with stacks from this end of the fragment, pair with stacks from this end of the fragment, as a measure of how well your assembly did, but also you can look at, for example, linkage between the 2 ends of your fragment, or throw out SNPs that are in linkage to equilibrium. And so from this, I had 2,500 or so polymorphic loci with about 23,000 SNPs, and that's with a 75% complete data set. About half from N1, about half from N2. About 95% of these map to the zebra finch genome, and about 75% map only once. And so this is a chromatogram of the ideogram of the zebra finch genome, and each black line represents where one of my SNPs are. And so first what I wanted to do was look at population structure. And so I threw in all of my frontalis populations, as well as my outgroups, the Guerrero subspecies, purple and Cassin's finches. And basically the purple and Cassin's finches fell out as one separate entity. The Guerrero house finch fell out as mostly another separate entity, and then the frontalis individuals fell out all together as one population. And we see the same thing in a discronant analysis of principal components plot. Sorry, I know we're running a little bit late. I'll try and speed through this. But what happens if the kind of more divergent populations may have been actually swamping out some of the variation that we see in the frontalis populations. So when I ran that by themselves, we actually get really nice definition between the eastern, the western, and the Hawaii populations, both in structure and with the discriminant analysis and principal components. Again, we can look at Hawaii, and interestingly, Kauai actually falls out by itself from Maui and Oahu. Both if you build a tree from it and in the structure analysis, and that has kind of interesting implications for how these birds were introduced to Hawaii. But, getting back to the point, what happened with genetic diversity is it actually decreased in these introduced populations. And so what I see is a 16% decrease in the Hawaii population, this is using the pi statistic, and a 7% decrease in the eastern population. So yes, there is a decrease, but not as strong as Dana Hawley had found with microsatellites and mitochondrial DNA. We see the same thing with observed heterozygosity and with private sites, so no matter kind of which statistic you use. I also looked at LD between my SNPs, and so this is on the same read. So between there's a distance of between 0 and 150, and we see elevated LD in the Hawaii versus the eastern versus the western population, which is something you would expect in bottlenecked populations. And also site frequency spectra do show the same thing. So again, we see more kind of intermediate minor allele frequencies than rare allele frequencies, which is something that you expect in bottleneck populations. Just for fun, we can look at, are there some FST outliers? And yes, we do see them. Now, it's hard to say if these are going to be because there was a selective event in these new different environments that they're living now, or if it's just due to neutral genetic drift during the bottleneck event. So I'm not going to go into that much further, at least at this time. Okay, so yes, there was some effect of the bottleneck, but not as strong as previously believed. But did the mycoplasma have any effect in the eastern population? And so I had sampling in 2001 from 3 eastern populations and the California population as kind of a negative control. So it's the same time period, but mycoplasma had not occurred there yet. And so did MG result in a loss of genetic diversity, and we detect signatures of selection in only about 8 generations. And so this is that pie chart just from the pre-epizootic subpopulations I had before. These boxes represent the post-epizootic, and there's no significant difference in the eastern population. So there was no overall loss in genetic diversity, even though about 60% of the individuals had died. What about signatures of selection? And so for the RAD data, what I wanted to do is I looked for FST outliers in my 3 eastern populations, but not in California population. And so for this particular analysis, I just look at the top 5% of FSTs that were in these 3, but not in California. And this is kind of hard to see, but there were 12 different loci that fit this criterion. 8 intergenic, 3 intronic, 1 intron/synonymous exon. 2 of these were near interesting genes. This ARAP2 is involved in T cell signaling, which is one of the major changes in gene expression between in that experimental study. This other one is involved in heparin and heparin sulfate synthesis, and heparin is actually used to treat conjunctivitis in people, which is one of the major symptoms of house finches and mycoplasma. So it's kind of an interesting gene too. So the conclusions here: Western, Hawaii, and Eastern populations show genetic differentiation and some loss of diversity in introduced populations. There's no loss in diversity in pre- and post-epizootic population comparisons, and there are potential loci under selection in populations affected by MG. And so, this is all very interesting, but for some reason, what I really want to look at is a few hard sweeps or many soft sweeps or that are actually responsible for the evolution of resistance and tolerance to mycoplasma. But leakage disequilibrium in house finches and other birds is very low. So in this paper, our postdoc in our lab, Nicholas, found that by about 1 kb, LD had almost completely dropped off. And I saw drop-offs in LD, you know, even within 150 base pairs in my own stuff. So you need much denser sampling than I had throughout the genome in order to actually think that you have any chance of detecting all the genes under selection. So now we have a house finch genome. We use one male individual from Arizona, which is a historically unexposed population. It's a pretty good quality genome, right up there with the highest bird quality genomes that are currently available. 1.12 gigabases estimated genome size, very similar to zebrafinch. Scaffold N50 of 2.2 megabases, contig of 71.0 kb, 70x coverage, and of the kind of core orthologous genes across vertebrates, only 4% of them are missing. So that's, again, right on par with some of the really high quality bird genomes that have come out. One thing you can do with the genome is throw it into PSMC and based on the coalescence of the two chromosomes in that single individual, make inferences about demographic changes through time. You can't get as recent a time as you would see with the bottleneck, but one interesting thing is we do see a huge increase in population size in house finches in general, not just the eastern population, about 1 million or so years ago. 300,000 to 1 million or so years ago. So right in line with the Pleistocene, which is something that North American ornithologists have been hypothesizing. And so with this sampling, I am going to be, at least for right now, sampling, the idea is 2 eastern populations in 1990, 2 western populations, and then so the disease spread through the east in 1994. I have these 2 populations sampled again in 2001 after the mycoplasma event, and then these 2 western populations plus 1 additional one that have never been exposed to MG. Then disease spread throughout the rest, and so now I'm going to have 2 populations that have pre and post epizootic in the west, 2 in the east, and 1 that's kind of a negative control. They've never seen the mycoplasma. So that's why I'm here this week, is collecting these individuals right here. I just got back like 3 weeks ago, my first population whole genome resequencing, and so that's from this Auburn population. I did 2 lanes of Illumina HiSeq 125 by 2, and I got about 334 million reads back. They're all of pretty high quality. This is just one example of a quality plot. And so for this I had 8 individuals from Auburn from 1998, from 2001, and 8 from 2014. This is kind of my play data set. Just with no filtering at all, I had about 27 million sites and average 2.7x coverage. This was just kind of a quick variant calling with mPileup mapping to the house finch genome. If I required variant sites to be present in 20 out of 24 individuals, I had about 11 million sites, which then averaged 3.5x coverage. So while coverage is low on an individual basis, when I'm thinking about it across populations, I have actually somewhat high coverage for the population itself. Now, I don't have too many more results from this to present to you today, but I just did a quick FST, and we do see a number of values. This is that same plot just blown up to the higher end of things in the high FST between 1995 and 2014. So I do have hopes that I will actually find some regions under selection. And so with that, I'll say I want to look at hard sweeps, soft sweeps, but also polygenic selection. And to do that, I can actually look at particular signaling pathways that I can hypothesize as being under selection due to the experimental infections, and look for overall FST changes. Finally, I'm almost done, I promise. What I can do once I find regions that are of interest, I have these birds that were experimentally infected from Arizona. From these I have phenotypic data like mycoplasma load and eye swelling score. This is a measure you could think of as tolerance. So individuals that have you can kind of standardize swelling to how much Mg you had. And I can actually test those regions that I found as being under selection in the geographic data. I can also look at MHC and TLR and see if they're actually associated with differences in phenotype experimentally. And finally, one thing I want to do is actually look at the evolution of immune genes across all birds, bringing back my whole house finch. And so what I find is really can be in the context of bird genome evolution. So with that, I have many people to acknowledge, my collaborators, my funding, and I am around all week. I would love to hear any feedback that you have or ideas.
It seemed like the historical range is like mostly Western North America, and then
Yeah.
so why didn't they ever spread eastward from the eastern edge of the range? Like they're able to come back west
So
from
the
east.
Rocky Mountains kind of block them off. They really are adapted to desert habitat, but because people in the east are so good at feeding birds, that's kind of what allowed them to get a foothold. So they're much more urban in eastern populations than they are in the west.
Okay.
You only really find them associated with humans. So they just have never been able to kind of make that jump over the Rockies and over. Yeah.
Other questions? Well, you're going to be around and able to answer questions as people have them this week. So where can we find you? Are you just working in the gallery? Or?
I'm actually down in the prep lab.
Okay.
But you can, if you send me an email, just Shultz at fas.harvard.edu. I'm happy to chat with you. Or, yeah.
Thank you very much. Yeah.